Hi everyone, welcome to another week of the Behavior Evolution and Culture Speaker Series. We meet every Monday at this time in this room and also with a Zoom option. And our speaker today is going to be on Zoom, but we're happy to have people joining us here to watch together. I'm going to just give you a quick preview of what we have coming up. All of the talks for the quarter are on the BEC website at bec.ucla.edu. And next week, Asher Rosinger is coming to speak also on Zoom. The title of his talk is Water Needs, Water Insecurity, and Nutritional Adaptations, the Intersecting Pathways to Human Biology. So we're looking forward to that. And in another talk that is going to focus on East Africa, we're really happy to welcome Andalia today. Her talk is entitled Lifestyle Change and Health in the Turkana of Northwest Kenya. She's joining us via Zoom from Vanderbilt University. And without further ado, I will pass the mic over to her and she can start speaking with us about all that's going on with the Turkana. Thanks.
Great. Yeah, well, thank you guys so much for having me. I was actually an undergrad at UCLA and I came to some of these talks when I was an undergrad, so it's fun to be on the other side. All right, so today I'm going to talk to you about my work looking at lifestyle change and health in the Turkana of Northwest Kenya. All right, so across the world, populations are transitioning from traditional subsistence level practices like hunting and gathering, subsistence horticulture, and pastoralism to what I'm going to call more Western, urban, or market integrated lifestyles. And these transitions are pretty universally accompanied by dramatic changes in physical activity, in diet, and in numerous other facets of human socioecology. So just to give a few motivating examples, first, here's some data from a review by Herman Ponser showing that the number of minutes per day that people spend engaged in moderate to physical activity is much higher in two subsistence level groups shown here in black relative to 6 different studies of urban or sort of high income countries shown here in gray. Just to give a little bit more motivation, here's data from a large study of US individuals run by the CDC, known as the NHANES study. And what you're seeing here is that for the average individual in the US, about 57% of the diet is made up of these really ultra processed foods that would never be available in a subsistence level setting. And so, not surprisingly, these dramatic differences in diet and in physical activity and in other facets of our environments that we observe between subsistence level versus urban groups are also associated with differences in disease risk, particularly in cardiometabolic disease risk. So for example, one of my favorite papers on this topic comes from the Chimane Health and Life History Project, which is a long-term study of the Chimane people of Bolivia who practice subsistence agriculture. And using non-contrast CT scans, Kaplan and colleagues measured coronary artery calcification. So this is like the amount of plaque that you have in your arteries in 700 Chimane adults. And what they found when they compared these coronary artery calcification scores for the Chimane versus other previous studies that had been done in high income settings, they found that the Chimane had very, very low levels of coronary artery calcification even into old age, right? So they have essentially undetectable levels even into old age. And that's relative to all these other studies where there's been very high levels, especially for older adults. Just to emphasize that the Chimane aren't necessarily special in terms of their cardiometabolic health, here's a summary of data from Eaton et al. Showing that type 2 diabetes prevalence across a number of modern day subsistence level groups, including hunter-gatherers, agriculturalists, and pastoralists, is about 10-fold lower than what we see in the US. And so again, these were just a few motivating examples, but I think this audience is probably pretty aware of the fact that there's now a large body of literature supporting the idea that both key features of lifestyle as well as many facets of health are consistently different between small scale subsistence level groups and urban, Western, or market integrated societies. And so as anthropologists and evolutionary biologists, I think we want to know, how can we interpret this? So why is it that living a small scale subsistence level lifestyle seems to be good for your cardiometabolic health, while living an urban or a Western lifestyle seems to be pretty bad? Well, one theory is that transitions towards urban lifestyles promote diets, activity patterns, and other types of environmental exposures that the human body is just not very well equipped to deal with. And in particular, there's a hypothesis from the field of evolutionary medicine known as the evolutionary mismatch hypothesis, which posits that traits that evolve in response to selection pressures in the ancestral environment of a population may no longer be advantageous if the environment shifts and individuals find themselves in a novel environment. And so with respect to cardiometabolic disease, the idea here is that humans evolved eating a mixed plant and meat-based diet that took considerable energy to obtain, and through evolutionary time, our metabolism and our physiology adapted to this type of diet and lifestyle. However, in Western societies today, we see a very different situation where a much higher in fat and carbohydrate diet can be easily obtained through a sedentary lifestyle. And therefore, it's been proposed that while subsistence level groups are more arguably sort of matched to the environments in which they evolved, Western groups are instead more mismatched, and that's what's leading to disease. And so this is a compelling and sort of intuitive explanation that's gained a lot of popularity. But I think that there's actually a lot of missing critical tests of this evolutionary mismatch idea. And that's not to say that I don't think it's probably true, but for it to be broadly useful for understanding modern variation in human health, I think there are a number of empirical gaps that we probably want to fill. So one of those gaps that I see is that the literature on these health comparisons between subsistence level and urban or Western groups is largely unconnected from the literature on local adaptation at the genomic level. So in other words, we don't have a lot of genomic evidence that subsistence level groups have adapted through evolutionary time to their traditional lifestyles, even though this is really a major and sort of driving assumption of the evolutionary mismatch hypothesis. Another issue is that the predictions of the evolutionary mismatch hypothesis are that individuals from a given locally adapted population will do better in the environment in which they evolved relative to a novel environment. But when we compare health between subsistence level groups versus the US or some other high income country, what we're actually doing is contrasting two populations that have different genetic backgrounds and usually very different recent evolutionary histories. And therefore, I don't think that we're really addressing how a given subsistence level group or the US individuals would perform in their ancestral environment versus a novel environment. Instead, I think we're just comparing two separate populations in a design where genetic background and environment are relatively confounded. And then finally, again, because sort of genomic work hasn't really intersected a lot with the sort of health and anthropological work that's been done in subsistence level groups, I think that we haven't made as much progress as we could on understanding how mismatch manifests at the genetic and the molecular level. So for example, we don't understand how molecular mechanisms like gene expression or other gene regulatory mechanisms might be connecting environmental effects with physiological and health effects that we see in the context of mismatch. And furthermore, we really don't understand what the genetic variants are that contribute to mismatch in ways that we would predict by the verbal model. And in particular, I think that under a mismatch model, what we'd expect to see is that diseases are driven by what's known as genotype by environment interactions. And so in other words, we'd expect there to be cases where there's some genetic variant that has different effects in the ancestral versus the novel environment. And there are a few different ways that I think this might manifest, but one possibility are these sort of cartoon reaction norms that I've put here on this slide, where there's a given locus that's usually associated with good health in the ancestral environment of the population. But when individuals move into a novel or an urban environment, there's some genotype that is associated with disease or with poor health. And so my research program is really focused on trying to chip away at some of these gaps using a range of different study systems. And most of my work in particular focuses on subsistence level groups in Kenya, Bolivia, and Malaysia that are currently experiencing rapid socioeconomic change toward more urban or market integrated lifestyles. And in each of these populations, I work with international multi-PI collaborative projects to try and collect environmental, biomedical, and genomic data that we can use to try to understand how populations have adapted through evolutionary time to their particular lifestyles. We then want to know how health changes as these locally adapted individuals transition to more urban or mismatched lifestyles. And then we also want to use the genomic data to try to understand what genetic variants and what molecular processes are actually connecting lifestyle change with health. And then a lot of my lab also is focused on complementing this field-based research with work in model systems. So namely, I do work with immortalized human cell lines as well as non-human primates. And both of these systems are more amenable to experimental approaches and as well as to the study of tissues that we can't sample in living humans. And today, what I'm going to focus on is telling you about work from the study population that is really the main focus of my lab, and that is the Turkana people of Northwest Kenya. All right, so first I want to just tell you about who the Turkana are. So the Turkana are one of the Nilotic peoples who are thought to have originated in the Nile Valley, so up in this region, around 15,000 years ago. And subsequently, they've spread out primarily throughout East Africa, and the red here shows the modern range. Within the last few thousand years, the people who identify as the Turkana are thought to have settled in Northwest Kenya, which is sort of circled here and then shown on the inset in this map. And since settling in Kenya, the Turkana have remained relatively isolated. And one of the reasons is because this particular region is extremely arid and inhospitable. So most of it looks like this year round. It's not a place that was sort of high on the list for colonization by the British, or where people have really sort of been wanting to move into. So in this region of Kenya, the temperatures year round are in the sort of mid to high 90s. There's very little rainfall on an annual basis, and this region is really prone to droughts if we skip one of these rainy seasons. As a result, there's pretty limited options for food or for water. So to cope with the water scarcity issue, the Turkana drink relatively little water each day, which they source from these really deep boreholes that they dig in dry riverbeds, and which they also walk long distances on a daily basis to obtain. And in interviews that we've conducted with Turkana, we've found that about 76% of people spend more than a few hours every single day collecting water, and 99% of the people that we've interviewed think that the water that they collect is insufficient. We've also used measures of urine-specific gravity to actually sort of empirically measure how dehydrated people are. And we find that 89% of people are going to meet the criteria for either being minimally or significantly dehydrated in this environment. Because this landscape is so dry and barren and water limited, it's largely unsuitable for foraging or for agriculture. And therefore, the Turkana people practice nomadic pastoralism, and they rely on livestock for subsistence. And as a result, they therefore consume a diet that's very meat and protein centered. So previous work by Galvin estimated that about 80% of the diet is coming from milk, from red meat, or from other animal products. And in interviews that we've conducted, we find that 74% of Turkana drink blood several times per week from their livestock. 97% are eating red meat several times per week, and 95% are drinking milk on a daily basis. And so this is what the diet and what the lifestyle of the Turkana people has historically looked like. And these are the types of ecological challenges that we think that they've adapted to through evolutionary time. However, over the last few decades, things have really started to change. So infrastructure has increased in this previously remote and isolated region. And that's the result of a number of complicated factors, but partly as a result of oil discoveries in South Sudan that have prompted a lot of road construction into the area. And so as a result of this, these changes, many Turkana have stopped practicing subsistence level nomadic pastoralism, and they're now living a more sedentary and market integrated lifestyle that's focused on trading items such as woven baskets, fish, or charcoal with the market economy that's sort of popped up around the increase in infrastructure. And in addition to the emergence of what I'm going to call this non-pastoralist group of people that still live within this relatively remote and rural Turkana region, there are also now some Turkana that have begun to leave both their homelands and their traditional pastoralist practices entirely, and are moving into central Kenya, where they experience a much more sort of densely populated, urban, and sort of Western-like lifestyle. And so about 6 years ago, when I was starting a postdoc at Princeton, I began a study of communities that span this lifestyle gradient. So particularly working with communities that still practice traditional pastoralism in the Turkana region, communities that have stopped practicing pastoralism, but again, still live within this relatively remote and rural region, and then communities that are living in central Kenya in these more fully urban contexts. And this study now has grown into what we call the Turkana Health and Genomics Project. So this is an ongoing, hopefully long-term study that's co-directed by myself, my former postdoc advisor, Juliana Rolls at Princeton, as well as Dino Martins at the Impala Research Center in central Kenya. And we're lucky to work with an amazing field team of local researchers. We also work with a great group of dedicated postdocs and grad students. And to date, we've worked with about 3,000 Turkana participants across 60 different communities throughout Kenya. In each of the locations where we work, we conduct in-depth interviews with our study participants. So we ask them about their diets. We ask about occupation and subsistence activities. We ask about modern amenities and possessions that might signal market integration to us. We do health and reproductive histories. We ask people where they've lived throughout their life, and we also conduct interviews about early life experiences. For each of our study participants, we also collect 9 different measures of cardiometabolic health. So we measure waist circumference, BMI, body fat percentage. We run a lipid panel, we measure blood glucose, and we measure blood pressure. And then finally, we also collect biological samples. So we collect whole blood, we isolate peripheral blood mononuclear cells, which is just the white blood cell fraction of blood. We collect serum, urine samples, and fecal samples. And before I tell you about the types of scientific questions that we've been asking with these data and samples, I want to say a few words about how we work with Turkana communities. So these communities are relatively hierarchical, and when we're interested in working in a given location, we first approach the chief and we discuss our project with him. If he thinks that his village might be interested, then we have a larger meeting with the local elders. And if that goes well, then we have a meeting with all community members. Before sampling, the study is also redescribed to each individual participant. And recently, my PhD student, Audrey Arner, has been working with a scientific illustrator to design some culturally appropriate animated presentations that we can use to support this process. But overall, sort of our strategy is to have the study explained at multiple different levels and give people many opportunities to ask questions. It's important to us that the communities that we work with benefit from the work that we're doing, and we therefore make community level contributions in each of the places that we work. We also always have local nurses on site that are working with us who can discuss abnormal test results or consult on any health related questions that community members might have. And then finally, we've recently done a lot of local training and capacity building through our local partner institution at Impala Research Center. And that's included building a molecular genomics lab that's available to local students and scientists who want to do genomic research in this area. All right, so using these data sets that we've built, what we've been trying to do is address these gaps in our knowledge about how evolutionary history and lifestyle change interact to affect health. And the first gap that we want to think about is the local adaptation piece. So specifically, we want to know how have the Turkana adapted to water stress and to pastoralism. So to answer this question, we generated 106 high coverage whole genome sequences, and we also generated 202 medium coverage whole genome sequences from Turkana individuals. And with these data, we then applied several different tests that can scan the genome to look for signatures of positive selection. So the first test that we used is something called the integrated haplotype score. And what this is going to do is to look for differences in the extent of linkage disequilibrium between 2 haplotypes at the same genomic position. So this test is based on the intuition that if there's some beneficial mutation that arises in the population and is under really strong selection, it's going to rise to high frequency really quickly, and recombination is not going to have enough time to break up that variant from the variants nearby. And that will result in sort of excessive LD on the selected haplotype relative to the neutral haplotype. The second approach that we used is something called the population branch statistic. And this is just a measure of genetic distance between 3 populations. And so specifically what we're going to try to do is identify loci where the Turkana have some large allele frequency changes relative to 2 reference populations. And in our case, we used publicly available data from 2 other African groups, the Luya and the Yoruba. And then the final test statistic that we're going to use is something called the X transpose X statistic, which is very similar in spirit to the population branch statistic, but it allows us to include an unlimited number of populations instead of just looking at 2 reference populations. But again, we're looking for sort of large allele frequency deviations relative to background expectations. So we ran all 3 of these different selection statistics that make different assumptions and use different subsets of the data. And we looked for regions of the genome that had a lot of outlier loci for all 3 of these statistics. And when we do that, we came up with 7 regions of the genome that are our candidate regions for positive selection. These regions fall near 7 different candidate genes that I'm showing you here. And there are many interesting genes in this list, including 2 that I've sort of highlighted here with asterisks that have previously been associated or have previously been found to be under selection in the Maasai, which is another East African pastoralist group. So that's really interesting. In the interest of time, though, I'm only going to tell you a little bit more about one of the genes from this list that we think is our best candidate, and that's a gene called STC1. So now what I'm doing is I'm zooming in on a 10 megabase region on chromosome 8, where I've got a genomic location on the x-axis and our evidence for selection from one of the statistics, the integrated haplotype score on the y-axis. So higher on the y-axis means greater evidence for selection. And so we can see that there's this clear peak of evidence here, and in the dotted line here is where the STC1 gene is. And in fact, the peak of evidence is just a little bit upstream of where the gene starts. And so since I'm only showing you this sort of snapshot of chromosome 8, one of the things that we should probably check is that the evidence that we see in this candidate region for selection is actually greater than the entire genome-wide background, right? And so that's what I'm going to show you here in this plot, where in black is the distribution of integrated haplotype score statistics for the entire genome-wide background relative to our candidate region here, and we can see that those are very different. One other way that I'm going to show you the data is to plot here something that's called a haplotype length diagram. And on this plot, each line is a given haplotype from a different individual, and the length here visualizes the length of the core shared haplotype. What we can see is that individuals that are carrying the derived, the putatively selected or beneficial SNP have these really long shared core haplotypes relative to individuals that are carrying the ancestral SNP, right? And I said that this is sort of the pattern we would expect if a mutation rises to high frequency really quickly. Okay, so hopefully that was convincing enough that there is some evidence of selection near this SCC1 gene. So now let me tell you what this gene does. So this gene has many functions, but it's most highly expressed in the final segment of the kidney, which is known as the collecting duct. So if you remember from your intro bio class, your kidneys remove waste and excess water from your bloodstream, and then they excrete the leftover in the form of urine. And this organ, and in particular the collecting duct, which is the terminal sort of portion of the kidney, are really, really important for determining water loss in the body. And this is an organ that's really sensitive to dehydration. In genome-wide association studies, this STC1 gene has been associated with glomerular filtration rate, which is a really commonly used biomarker in biomedicine for how well your kidneys are functioning. It's also been repeatedly associated with uric acid levels, which are waste products that are produced by the kidneys when the body breaks down purine-rich foods, such as red meat. In rodent experiments, STC1 has been shown to be involved in glucose homeostasis, such that if you infuse human STC1 into the medulla region of the kidney, it alters glucose production. And then finally, STC1 has also been shown to be involved in the response to dehydration in rodent experiments, such that if you experimentally dehydrate a mouse, expression of this gene shoots up over a 72-hour period. And so this is all really interesting to us, given that we know that the Turkana eat a diet that's really high in red meat and high in purines, and they also are facing very constant dehydration and kidney and water stress. And so the next thing that we wanted to do was to more functionally link some of these phenotypes to STC1 in humans, since the work that I just described to you had all been done in mice. And we also wanted to link STC1 genotype to phenotype in the Turkana themselves. And so to do that, we performed 2 different experiments. So the first thing that we did was to take a human kidney cell line and treat it with antidiuretic hormone. So this is a hormone that's secreted during dehydration, and it tells the collecting duct to just hold on more tightly to water. After treating the human kidney cells with this antidiuretic hormone, we then performed qPCR and confirmed that, in fact, the STC1 gene is sensitive to this dehydration signaling in humans. The second experiment that we did was to use serum samples that we collected from Turkana study participants. And we then used those serum samples to measure two different biomarkers of kidney function. In particular, we measured urea and we measured creatinine. And we did this across about 450 individuals, and we also generated genotype array data for those same individuals. And using those data sets, then what we wanted to do was to do some really focused association mapping to see whether we could link genotype to phenotype within this candidate STC1 regulatory region. When we ran those analyses, we didn't find any associations for creatinine, but we did find an association that passed our multiple hypothesis testing thresholds for urea levels. So this is the STC1 gene, and this is the SNP that we find to be associated with urea levels. And here's just what that looks like over here, where I'm showing on the bottom genotype, and on the y-axis urea concentration for individuals with the three different genotypes. And so there's still a lot of sort of ongoing work here and a lot more that we need to do, especially to sort of fine map the causal SNPs and really figure out precisely what the physiological changes are that's going on. But I think it is really exciting that we have been able to link this human dehydration response to the STC1 gene, as well as to link genotype to phenotype within our candidate regulatory region. And I'm just going to point out that, I only sort of touched on a subset of this story, but if you're interested, we have a preprint up on bioRxiv, and you can read more about this work. All right, so now we've got some evidence that the Turkana are locally adapted to water stress and to pastoralism. And so now we want to know what happens when we take individuals from this locally adapted genetic background and move them into a very different environment, move them into an urban or sort of Western-like environment. All right, so to ask this question, we're going to turn to the lifestyle gradient that I described to you earlier in the talk. And in particular, the analyses that I'm about to show you are from a study that we did focusing on the cardiometabolic health biomarker data collected across these three different lifestyle groups, from traditional pastoralists, individuals that have stopped practicing pastoralism but still live within the remote and rural Turkana region, as well as individuals that have fully transitioned into an urban lifestyle. And again, this is about 1,200 people, but some of the biomarkers have smaller sample sizes, and that's shown in this table. But before I actually sort of show you what the health comparisons look like, comparing these three different lifestyle groups, I first just want to show you some of the metadata and the interview data that we've collected to convince you that these three lifestyle groups really do follow a gradient from sort of traditional and more, matched to the evolutionary history of the population to more urban and mismatched to that ancestral lifestyle. And so that appears to be true if we look at one commonly used measure of urbanicity, and that's distance to a major city across the three lifestyle groups. So we can see that pastoralists are very far from any major city. Urban individuals are very close, and the sort of intermediate group is falling in between. In addition, if we look at the dietary items that people say that they're commonly consuming within these three different lifestyle groups, we can see that almost all of the traditional pastoralists, which are shown here in light blue, report that they regularly consume blood, milk, and meat, as would be expected, and they're consuming very few carbohydrates and very few food additives that would be obtained from the market economy. In contrast, individuals that are living in urban areas report that they consume, fewer animal products. They're consuming a lot more carbohydrates and a lot of food additives. And individuals that are not practicing pastoralism but still living in the rural area are shown here in dark blue, and they sort of fall as an intermediate between these two extremes. All right, so now let's look at how health differs between each of these three lifestyle groups. I'm about to show you a plot where we're going to look at each pair of lifestyle groups against each other. On the x-axis, I'm going to show you the standardized effect size for each of those particular contrasts. And if you see a larger effect size, that indicates that there was worse health in the more mismatched or urban group. If you see a translucent bar, that means that the particular contrast of interest was not actually significant. And here's what the results look like for all of those analyses. So what we can see is that we do see consistent health differences between pastoralists and urban individuals. So those are the contrasts shown here in blue. We also see consistent health differences between rural individuals that are no longer practicing pastoralism versus urban individuals. So those are the contrasts shown here in green. But we really don't see any health differences between pastoralists and the rural individuals that have stopped practicing pastoralism. So those are the red bars here, which are all translucent, meaning that none of them were significant. And so just to summarize what I think this is telling us is that we do see health changes in Turkana that are living increasingly urban lifestyles. However, these effects appear to be nonlinear, such that we're really only seeing evidence of increased cardiometabolic disease risk in individuals that are living this fully urban lifestyle. And for the sort of intermediate group, we're really not seeing health variation that seems to be all that different from the subsistence level pastoralists. Just to give you a little bit more context about the magnitude of the biomarker differences that we're seeing and their sort of potential consequences for disease, what we did was to take cutoffs from the CDC and the American Heart Association that are used to diagnose diseases like hypertension, metabolic syndrome, or obesity. And we just asked how many individuals in the urban lifestyle group actually meet these criteria for disease relative to individuals within the two rural lifestyle groups, which I'm now just going to group together for the remainder of the talk because we didn't really see any health differences between them. And then on this plot, I've also put data for the US from the NHANES study just to sort of give you a frame of reference. And so what we can see is that for urban Turkana, we are definitely seeing individuals that would reach the thresholds that would sort of put them at risk of cardiovascular disease. We actually see a little bit of that as well in the rural Turkana. None of this is reaching the levels that we see in the US, but it is sort of signifying to us that these urban to rural transitions in Turkana are not just creating variation in health biomarkers, they're actually sort of putting individuals potentially at risk. All right, so now I've got some evidence that the environment that you are currently living in, sort of broadly grouped into urban versus rural, seems to be important for your cardiometabolic health. But a question that I have always been interested in and really continue to be very interested in is, does the developmental environment matter for these adult health outcomes? So all the data that we've been collecting is from adults, but we might actually want to know whether it matters whether someone just recently moved into this urban environment versus whether they've spent their entire life there, right? So how does the developmental environment play into this? Does the early life environment matter for explaining adult health outcomes? And it's possible for us to ask this question within the Turkana population because there's so much within lifetime movement right now. So for example, here's data from just one individual in our study showing their birth location here in red, connected to the location where we sampled them as an adult by this gray line, and the adult sampling location here is the black outlined dot. And so what we can see is that this person was born in a very different environment relative to the one where we sampled them as an adult. And so now I'm putting all of the data on this plot for many individuals in our study. And again, what we can see is there's just a lot of within lifetime movement, including there are many people that were born in rural areas and moved to urban areas, as well as some that go in the opposite direction. And this movement means that the early life and the adult environments within our study are not entirely confounded, and therefore we can try to statistically sort of tease apart their independent effects. And in particular, for the analysis that I'm about to describe to you, what we did was to ask whether the population density of each individual's birth location, estimated during their birth year, predicted their adult health controlling for adult lifestyle. And just to drive home this point that I've been trying to make about the fact that early life and adult conditions are actually undistinguishable, here's a plot just showing that early life variable that we're going to use broken down by individuals that were sampled as adults in urban areas or in rural areas, right? So we do see, consistent differences, but there's also a ton of variation within the adult urban group as well as within the adult rural group. And so now we're going to try to link this early life variation to adult health outcomes. And so when we ran these models, we found that there were significant early life effects for 4 of our biomarkers. So here's what the raw data look like for those 4 biomarkers. So on the x-axis, I'm plotting the log 10 population density for each individual estimated, or for their birth location estimated during their birth year. And on the y-axis are these biomarkers measured in adulthood. And what we can see is that being born in a more urban or high population density location is associated with higher BMI, higher body fat percentage levels, higher waist circumference, and higher blood pressure. And I think that this result is really important and really interesting because it suggests that there are, in fact, independent effects of early life and adult experiences. I think it also suggests that cumulative exposure is going to be what really matters for people, and the individuals that spend their whole life in an urban environment are going to be those that are at the greatest risk for cardiometabolic disease outcomes. All right, and I also do just want to say that, you know, while the Turkana Health and Genomics Project was originally really focused on looking at lifestyle effects on cardiometabolic health, because of the STC1 work, we've also recently become very interested in thinking about lifestyle effects on kidney health. And so, in fact, from our preliminary data that we've generated from serum, urea, and creatinine measures for those 447 individuals, we actually sort of broke this down by individuals that are still practicing pastoralism in the rural areas, that are not practicing pastoralism but still living in the rural areas, as well as urban individuals. And we found that there were significant differences, particularly there's higher creatinine for individuals that are living an urban lifestyle, and there's higher urea levels for individuals that are practicing pastoralism. And so one of the sort of major thrusts of the project right now is that we recently got some machinery that will allow us to use ultrasounds to actually measure kidney size and look at kidney structure and function. And we want to know how lifestyle is affecting kidney health. And this is something that we're really excited about. All right, so now we've talked about local adaptation. We've talked about what happens when locally adapted populations move into very different environments. And now we're left with this last problem of not really having a great handle on the molecular or the genetic mechanisms that might be connecting lifestyle effects to health in the context of mismatch. And so this is a really important question to me because a lot of the work in my lab is focused on how changes in gene regulation might be a mechanism that connects environmental inputs with health-related traits. And so in particular from work that I've done, but also from work that many other people have done, there is now at this point a lot of evidence that when we see variation in environmental variation that's connected to variation in health, that what's often going on at the molecular level are that there are changes in epigenetic gene regulatory mechanisms, for example, changes in DNA methylation. And so DNA methylation is the addition of methyl groups to cytosine bases, which in this cartoon I'm just representing as filled in lollipops here, so these little dark circles. Variation in DNA methylation often sort of clusters in promoter regions or in regions of genes that are really important for actively regulating the expression of the gene. And so there have now been a number of studies that have been able to link environmental variation with sort of environmentally driven differences in DNA methylation. Those differences in DNA methylation can in some cases then go on to affect the way that a given gene is expressed. Usually this goes in the direction that if we have high levels of methylation, for example, in the promoter of a given gene, that usually leads to lower levels of expression, whereas low levels of DNA methylation are usually linked to higher levels of gene expression. And then finally, those differences in gene expression can then scale up to influence physiology and to influence health. And so we're interested in testing in the hypothesis that these cardiometabolic health differences that we see between urban and rural individuals, and again, I'm just going to group pastoralists and sort of the other subsistence level rural individuals together here. We want to know whether these differences in health that we're seeing have anything to do with gene regulation, so with DNA methylation or with gene expression. And so what we did to try to test this hypothesis was to use the blood samples that we had collected from our Turkana study participants. We used these blood samples to measure genome-wide gene expression. And for the genome-wide gene expression work, we specifically measured this from peripheral blood mononuclear cells. So again, that's the white blood cell fraction of blood. And then for the DNA methylation data, we used whole blood to measure DNA methylation using a technique called reduced representation bisulfite sequencing. In total, we were able to measure 452 transcriptomes and 329 epigenomes. The gene expression data covered about 9,000 genes, and the DNA methylation data covered 650,000 CpG sites. To do these these analyses in like what I would consider the proper way, what we really want to do is control for different covariates that might be important for influencing gene regulation, and that's namely sex, age, cell type composition, and genetic structure. And right now, we don't actually have all of the data that we need to analyze these covariates for the full data set. And so right now, I'm going to show you preliminary analyses looking at about 2/3 of each of the data sets, but we're working to build out the rest of it. And once we do, then we'll hopefully be able to analyze the total data set. All right, but let's just start by looking at sort of what we got right now. And the first thing that I want to do is compare DNA methylation patterns between the rural and the urban individuals. So to do this, what we're going to do is to run a statistical model for each one of those 650,000 sites that we measured. And if we run these 650,000 different tests, and if there was no genome-wide signal, what we would see is just basically randomly distributed p-values, right? So we would see that any p-value between 0 and 1 is equally likely, and we would get this, this uniform distribution if there was no genome-wide effect. However, when we actually run our analyses genome-wide, that's not what we see. Instead, we see an enrichment of low p-values indicating a global effect, right? So we see more tests that have low p-values skewed towards 0. If we correct for multiple hypothesis testing, we find 267 sites that are differentially methylated at a 10% false discovery rate threshold, and we find more like 4,000 sites if we relax this threshold a little bit. All right, so now let's do the same sort of analysis for the gene expression data, just to get a feeling for the global data set. So now, again, we're going to run a statistical test for each one of our genes. We've got about 9,000 genes. If there wasn't any signal, then we would expect this uniform distribution, but that's not what we see in our observed data. Again, we see a skew towards low p-values indicating a global effect. And if we correct for multiple hypothesis testing, we are able to identify 545 genes that are differentially expressed between urban and rural individuals. All right, so now we've got some evidence that there are sort of global genome-wide associations between lifestyle and DNA methylation, as well as lifestyle and gene expression. And so the next thing that we want to do is to put these results together and ask whether the lifestyle-associated changes in DNA methylation are actually sort of regulating or driving the differences in gene expression. If this were true, then we would expect there to be a correlation between the effect sizes from the two analyses. So in other words, we would expect that for genes where we see an increase in DNA methylation driven by lifestyle, that would be met with a corresponding decrease in gene expression at that same gene, right? And presumably this relationship would be really strong for lifestyle-associated genes. But if we looked just at random genes or genome-wide, then we wouldn't expect to see this type of relationship. And so when we run these correlations, that's exactly what we see in our data. When we look genome-wide, we don't see any relationship between the lifestyle effects on DNA methylation and on gene expression. But if we look at lifestyle-associated genes, we do see this expected negative correlation. All right, so now we know that lifestyle affects methylation, potentially in ways that are impacting gene expression. And so now let's try to actually dig into the biology of what these differences in gene expression are telling us. So the first thing that we can do is to look at the direction of the differential expression that we're seeing. And the way that I'm going to show that is by what's known as a volcano plot. So what I'm showing you here on the x-axis is the effect size for the lifestyle effect. And on the y-axis is the magnitude of the minus log 10 p-value, such that genes that load really high have the most sort of significance. And the genes that pass our genome-wide multiple hypothesis testing correction are colored here in blue. And so what jumps out at us from looking at this volcano plot is that there are a lot of genes that are sort of going in one direction, right? We see a lot of genes that have higher expression in the rural individuals relative to the urban individuals. And so now we want to know what the heck these genes are. And so to do that, we use a technique that's called gene set enrichment analysis, which allows us to just annotate all of these genes for the different biological pathways or processes that they're involved in. And when we do that, we get a whole mess of stuff, but let me just sort of walk you through this a little bit. So one of the clusters of genes that we find here involves a lot of genes that are involved in biological pathways such as response to heat or cellular response to heat, or different processes that cells do when they're sort of under heat stress. And that, makes a lot of sense to us, given that I told you that the Turkana are under this constant heat stress and sort of dehydration stress. Another big cluster of genes that we get here are those that are involved in the immune response. And this is actually sort of most of the genes that we see in this particular group. So we get genes that are involved in pathways like cellular response to cytokine stimulus, cytokine-mediated signaling, or response to chemokine. And this also actually makes a lot of sense to us, given that we know that the pathogen environment is very different in the rural areas relative to the urban areas. For example, from interviews, we know that people have much more malaria and helminth burdens in the rural areas relative to the urban areas. Another way that we can think about this immune involvement for the genes that we find to be lifestyle associated is to use some previously generated data. So what we did was we downloaded lists of genes from a previous study that actually took the same cell type that we're using and exposed the cells either to LPS, which mimics a bacterial infection, or to Gardaecumide, which mimics a viral infection. And then we took those lists of either bacterial or viral response genes and overlapped them with our genes to see whether they were significantly enriched. And we find that that's the case for both the viral response genes and the bacterial response genes, and a little bit more so for the viral response genes. All right, so now we've got evidence that these rural lifestyles lead to increases in gene expression, particularly at immune-related genes. And I told you from previous work in the study population that we've also linked this rural subsistence level lifestyle to better cardiometabolic health, so to decreased cardiometabolic disease risk. And so the next question that we want to know is whether, are these independent associations, or is there actually any evidence that these differences in gene expression that we're seeing might actually be connecting lifestyle variation with variation in health? And so we tried to get at this in a few different ways. One approach that we took was to overlap our list of differentially expressed genes with lists of genes that have previously been identified as being sort of causal at the transcriptional level to 114 different human diseases. So for each of 114 different human diseases, we have a list of genes that people think are causal at the transcriptional level, and we can overlap that with our lifestyle-associated genes and see whether there's any enrichment. And so when we do that, we find, not surprisingly, that a lot of our genes seem to be involved in autoimmune disease phenotypes. But the diseases that are actually most strongly enriched among our lifestyle-associated genes are, in fact, cardiometabolic diseases. So namely, cardiovascular disease is very strongly enriched, as well as type 2 diabetes. All right, so that's pretty interesting, but it's still, a little circumspect, not necessarily direct. And so we want to test this idea more directly. And to do that, we can use a technique that's called formal mediation analysis. So this is a statistical approach that allows us to ask whether the lifestyle effect on a given health biomarker is entirely direct along pathway number 1, or whether when we take gene expression into account, there's actually a modification of the direct effect, which would indicate mediation. And so I'm not going to go sort of too deep into the weeds about how these formal mediation analyses work, but they're a relatively sort of established way for trying to test sort of different pathways along a causal diagram. And we did these analyses focusing particularly on BMI, body fat percentage, and waist circumference as our health outcomes. And we did that because these biomarkers we knew were associated with lifestyle, and also because we had the largest sample sizes for those particular ones. And so we ran these analyses, these formal mediation analyses, for our 500 differentially expressed genes. And when we do that, we find that anywhere between 40 and 70 of those 500 genes show evidence for significant mediation. So that means that there is indeed some evidence for this purple pathway number 2, but there's also a lot of genes for which the relationship between lifestyle and expression is pretty independent of the relationship between lifestyle and health. And so you may be scratching your head here a little bit about the fact that I'm telling you that we're seeing increased immune gene expression related to decreased risk of cardiometabolic disease. Since in the industrial world, or sort of in the Western world, people tend to think of this relationship as going in the opposite direction. So usually we think of inflammation or sort of upregulation of the immune response as being associated with worse cardiometabolic health outcomes. But I think that there's actually some precedent for thinking this relationship might work differently in a subsistence level context. So I'm going back now to the Chimani population that I talked to you about earlier in the talk, who again have the lowest measured rates of coronary artery disease in the world. However, the Chimani also live in the tropics. They're heavily parasitized, and they have off the charts levels of immune activation relative to Western societies. And in fact, this same study that I was talking to you about from Kaplan et al. Actually looked to see whether the different immune biomarkers were associated with cardiovascular health outcomes. And they actually found that there was lower disease risk associated with higher biomarkers of immune function. So higher IL-10, IL-5, and different higher levels of white blood cells. So this is consistent in direction with what we see in the Turkana, and again, is very opposite to what people usually observe in Western settings. And so I think that this really, paints a picture that there might actually be some degree of immune activation that's protective against cardiometabolic disease risk in a subsistence level context, even though that doesn't really seem to be how it works in the Western world. All right, so I just talked to you a little bit about molecular mechanisms, namely DNA methylation and gene expression that might be involved in mismatch and lifestyle effects in the Turkana, but I didn't actually just say anything to you about genetic mechanisms. And in particular, I said earlier in the talk that I think that under the verbal model of evolutionary mismatch, that what we'd really expect to see are these genotype by environment interactions, such that there are loci that are actually sort of beneficial to health in the ancestral environment of a population. But then in a novel environment, those same loci become associated with disease or with poor health. And I think that this is really one of the key missing empirical pieces of data that we need to validate and to understand mismatch in humans. However, mapping loci that look like this, or just mapping genotype by environment interactions in general in humans, has been extremely difficult. And so it's been difficult for a number of reasons. One is that you need really large data sets. You need a lot of power to map interaction effects. Another issue is that the right environmental comparisons are often difficult to define and to assemble, right? So to do what I'm showing here on this slide, we need genomic and phenotypic data from people that are living in two very different types of environments. Even if we could actually do this, there's usually going to be a lot of confounders if we're doing this in human populations. And the studies that I think have made progress to date and that have had really large sample sizes, sort of had the right composition of data types, have so far been really medically focused rather than evolutionarily focused. And so even though there has been some progress made on G by E interactions, no one's really been looking at it as much as I would hope that we would in a mismatch framework or sort of from an evolutionary framework. And so how the heck can we make progress on this this really important problem? Well, I think one approach that we can use is to use experimental systems where a lot of these issues that I just laid out are relatively solvable. And so I'm not going to go into these studies, but I do just want to plug two recent papers where my co-authors and I tried to explicitly test for genotype by environment interactions in the context of mismatch using both fruit flies as well as human immortalized cell lines. And in both cases, we do find a lot of evidence for genotype by environment interactions that are consistent with mismatch. And I also do want to say that I don't think all hope is lost on trying to do this sort of work in humans. And I think that one path forward that I'm especially interested in pursuing is that I think that we could learn a lot by bringing together studies from many different populations that are all undergoing lifestyle change. So at this point in time, there's a lot of work being done across the world by anthropologists, geneticists, physicians, trying to understand how lifestyle change is influencing health. And I think that if we sort of all work together, there might be some commonalities and some power gained. And at least that's what we sort of argued in this working group of folks that we got together to talk about this really key issue. All right, so let me just summarize what I told you, and then we can move to the Q&A. So I showed you some whole genome sequencing data that we use to uncover evidence of local adaptation near this gene called STC1. This gene has previously been linked to purine metabolism, kidney function, and dehydration in rodent models. We were able to show that STC1 is sensitive to dehydration signaling in humans, and also that there are some genotype biomarker associations at this gene in the Turkana themselves. And again, if you're interested in sort of learning more about this story, we have a preprint up on bioRxiv. I then told you that when these locally adapted Turkana individuals move to novel urban environments, that their cardiometabolic and potentially their kidney health starts to suffer. Some of these lifestyle effects on health are associated with differences in genome-wide DNA methylation and gene expression, and some of them seem to maybe be involved in mediating the lifestyle effects on health, though there's also a lot of sort of independent effects that we're seeing. Ongoing work for the Turkana Health and Genomics Project and for my lab is really focused on trying to understand kidney health in the context of evolutionary mismatch. I have a lot of personal interest in looking at early life effects on adult health, and that's something that we're really trying to develop and expand out in the Turkana. And then I'm always interested in trying to get more evidence for genus height by environment interactions. I'm actively recruiting grad students and postdocs, so if any of these topics interest you or if you know anyone, please send them my way. And with that, I want to thank again all of the team members for the Turkana Health and Genomics Project. This is a highly collaborative group of folks, and none of what I just presented is me alone. It's all really team science. I especially want to thank my co-directors, Juliana Rolls and Dino Martins, as well as our field team and the postdocs and grad students that work with us. I want to thank all of the study participants and the communities that we work with, who we've learned so much from, and without whom, none of this would obviously be possible. And then I want to thank additional collaborators. We've got collaborators and support staff. So we have great support staff at the Impala Research Center in central Kenya, collaborators at the University of Nairobi, Princeton, UCSB, Arizona State, that have all been involved in especially the natural selection work. And then I want to thank my lab at Vanderbilt, as well as my funding sources. And with that, I can take any questions.
Great, thanks, Amanda.
Right.
I'm going to just be on for hands if there are questions.
Okay. All right, yeah, I'm going to stop the share so that I can see you guys better, but I'm happy to go back to any slides if there's anything that would be of interest.
While we're waiting, I just have a quick question about food security, and just did that differ significantly between the rural and the pastoralists? Just thinking about the sort of proximate pathways of some of this.
Yeah, it definitely does. Well, I guess, do you mean within the rural areas between pastoralists and non-pastoralists? I'm
thinking about that sort of middle group, right? Because you wound up lumping them together because it didn't look like there were very many differences. But I was just wondering if there were things that you picked out from either food or water security that seem to be changing as people start to make the transition, presumably toward a more kind of urban lifestyle.
Yeah, there definitely is. I mean, both of those groups are still having a lot of water and food insecurity, but I would say that there's greater food and water security in the the non-pastoralist group, given that pastoralism is just so tied to like what's going on with the ecology, right? And there's currently a huge drought in Kenya, and that's been really, really difficult for the pastoralists, especially. I'll also say that, for that study that we didn't find significant differences between the sort of intermediate group and the pastoralist group. But as we've started to get larger and larger sample sizes, we are starting to see some small differences. So I think that it's probably not that there are no differences, it's just that they're relatively subtle relative to the urban group, and we just need sort of bigger sample sizes to be able to detect them. Great, thanks. I see Molly's hand up. Yeah.
Oops. Amanda, it's such a fascinating, inspiring talk. Wow, it was so cool. Thank you so much for sharing.
Thank you so much.
Wow, I'm blown away by everything you've done. I have a really simple question, just wondering, because you've obviously done so many different analyses. I thought if there's anything you hadn't shared on this, the effect of being born in one locale, but then being an adult residing in another. Leaves a lot of like unexplained life experience between the moment of birth and the moment of testing, I guess.
Absolutely.
And I was curious if you had any analyses or thoughts about, I guess, critical periods or sensitive periods, or whether you think, was there some sort of prenatal effect, especially because it's cardiometabolic outcomes you were looking at, and birth is actually reflecting prenatal, not postnatal exposure. So yeah, I was wondering anything about that.
Yeah, no, that's a great question. Yeah, so that's where I'll where I sort of see the next like 5 to 10 years of my research really being focused. So for that initial study, we only had asked people like where they were born. So we just had, like a GPS point, and that's why we were using population density of the birth location. Since then, we in the last like 2 years or so have started doing a pretty in-depth interview with people about their early life experiences. So this is, everything about like sort of what was subsistence like for your family when you were growing up. We've also sort of adapted the adverse childhood experiences survey that's used by the CDC, tried to adapt it to Turkana's. So the point being that we, now have like 50 questions that we ask people about their early life experiences, and we're trying to get a much better handle on sort of what is it about early life that is different as people's lifestyles change, and what about that is important for health. So I don't really have an answer to your question yet, I guess, but the point is like we're working on it. Give me a couple more years, and hopefully we'll know something.
Cool, thank you.
Kim?
Hi, thanks so much for this talk. It's really great.
Thanks.
I was wondering if you have been looking into how the genetic variation in your population shapes patterns of methylation that respond to these different lifestyles.
Yeah, that's a great question. We have not dug into it too much yet, but that's definitely on my list. I think that is also a place where I'm very interested in how genetic variation influences DNA methylation, and I'm also interested in using methylation then as sort of like an endophenotype that we could use to look at G by E, right? Because then we could ask, are genetic effects on methylation different between the different lifestyles? So I guess that this is another area where I guess the answer is not yet, but yeah, I think that's a great question, and something that I'm hoping to get a student that's interested in doing that.
Ryan?
Fantastic talk. It was a wonderful, wonderful presentation with so much great information and results brought together so nicely. So just want to echo everyone else. Thank you for your
Thank you.
And I was also wondering, what with my work in the Hadza, focusing on cardiovascular disease risk indicators, and I'm sure you've encountered this too in your work. People generalize from one domain of health to overall well-being. Frequently, people will say, oh, well, their CVD risk is low, so that means they're healthier in general. And I have to push back against that quite a bit, especially given some of the more gross measures of health that one would consider in a kind of human biology or well-being framework, including just overall demography, like mortality rates in particular. So I'm wondering if you have enough samples in your study thus far to be looking at things just like mortality risk, or related to this issue of what happens when you move into urban environments, also changes in fertility that might be occurring in these subsets of your group that you're focusing on, and just to kind of step back and put things in terms of fitness rather than a more detailed measure of health.
No, that's a great. Yeah, that's a great question. So we've got some data on these things, and again, like trying to collect more, but I can tell you what we know so far. So we definitely don't have enough data about, although like we have no data really at this point about adult mortality. We have started collecting some data on infant mortality. This is, and to be fair, this is all entirely like self-reported. Like we do interviews with people about sort of how many of their kids have died and at what ages. And so I can tell you that in the pastoralist groups and in the rural areas in general, infant mortality is really, really high relative to the urban areas. So that's probably not terribly surprising to you, but that is a pattern that we definitely see. We also know that female maturation happens earlier in the urban areas, and that fertility is lower in the urban areas, and that seems to be driven by sort of family planning, like there are more people that are on birth control and women are waiting longer to have kids in the urban areas. So so definitely from a fitness point of view, right, the story is much more complicated. Was there something else that demographically that you asked about that I did not touch on?
No, that was it.
Yeah. Okay.
You hit fertility and fertility. That's it.
Yeah. Yeah. I'll also say, I mean, another thing that I can throw out there is that we also do, we ask people about their self-reported health, right? So like there's a difference between like, is your blood glucose high, but like, how do you feel, right? Like, do you feel like your health is good? As well as, you know, we sort of ask people about, do you have like headaches, or does your back hurt, or, you know, like they have like a detailed sort of self-reported health survey. And there are a lot of you know, sort of experienced aspects of health that are not good for people that are practicing pastoralism. And so, yeah, so I definitely sort of tried to simplify the story about cardiometabolic disease risk. But if we think about fitness, or we think about, you know, people's actual like experiences of health, that's a much more complicated picture for sure.
Thanks.
Yeah. All right.
Dan?
Dan.
Thanks, Amanda. Really, really interesting. I find it a little hard to believe that you, as an undergraduate, attended Beck Talks, because that would mean that I'm really old, that you've done all this fantastic work. But so be it.
I'm also getting old.
That doesn't help me. So this actually, Brian set me up nicely for this one, or your discussion with Brian, which is, what is the source of balancing selection on things like STC1 in populations that aren't exposed to this, you know, extremely arid environment? This is question one, just kind of simple question. And question two is, and I know that you just kind of briefly touched on this towards the end, but again, it fits in with the portrait of overall fitness. You said that you had fecal samples, and I'm curious whether you see in some of those areas associated with immune activation, whether you see, you know, beneficial effects of helminth infection in, you know, in the rural populations relative to the urban.
Yeah, those are both very good questions. So we only recently started actually generating like objective, you know, doing fecal floats and sort of measuring helminth burden. And so right now I can only tell you that it's higher in the rural areas relative to the urban areas. We haven't tried to correlate it with anything beyond that, but hopefully we'll get there. This was sort of it took us a while to figure out how to do this well at a large scale. So hopefully coming down the pipeline at some point. And then for the question about like balancing selection and STC1, that is a really great question that like keeps me up at night. So I didn't go into all of the details about STC1 just sort of in the interest of time, but the SNP that we find that has the strongest evidence for selection, which like in no way am I claiming it's the causal SNP, but like let's just say, you know, we've got a SNP that's got a lot of evidence. If we look at the distribution of that SNP genome wide, it's actually at like 2% allele frequency outside of Africa. So this SNP is like really either being selected against or was just really lost in the out of Africa bottleneck, right? Like I don't know, but it's really, really striking that it's at sort of low to intermediate frequency in non-nilotic African groups, and then it's at really high frequency in the Turkana, as well as we've sequenced some other nilotic groups that also practice pastoralism sort of in similar environments. It's at high frequency in those groups, and so yeah, I would love to know the answer of like why is it low elsewhere and high in the Turkana, and has it not gone to fixation because there is some sort of like some sort of cost, or is it just that there's been sort of relatively recent selection on this variant and therefore it just hasn't quite reached fixation yet? So all good questions. I suspect that there is some sort of cost of this allele or something else that's going on, especially given that, like I said, it's at like outside of Africa, it's essentially like not invariant. Like in some populations, it's at less than 1% allele frequency. So it's, yeah, really interesting.
Okay, and fascinating. Thank you. Can I just throw this out there as like this wild speculation that you
Oh,
might
please.
be
Yeah.
able to address in some of your work. So there's, and I haven't kept up with this literature recently, but there was previously work showing that prenatal and early life dehydration had a lifelong impact on salt preferences. It upregulated salt preferences, and this is demonstrated in Western populations. I'm curious whether you see whether you're the kind of dietary information that you're collecting either currently or potentially in the future could explore that, and whether there is any link to exposure to arid environments and gene regulation accordingly. Because I had a wild speculative hypothesis that this was functional that I published years ago, and it seems like this is a unique opportunity to potentially explore the topic.
Cool. Yeah, no, that was not something that had been on my mind, but that sounds, yeah, that sounds very up my alley. That's great. Thank you.
Thanks.
Is there questions? No? Give one more minute. People on Zoom. Okay, that's awesome. Thank you so much, Amanda. It was such a great talk.
Yeah.
I really enjoyed
Thanks.
having you. And we will see you all here for back again next week.
All right. Yeah. Thanks, everybody. Thanks so much for coming. All right. Have a good rest of your Monday, everybody.
Thanks. Bye.
Thanks, Amanda.
Bye.
Bye.